Hello, my name is Lisette Molins and I'm part of the Marine and Coastal Biology Lab of Universidad de Carabobo. And I'm here to talk about one of our projects, which is polychaetes associated to Ircinia felix on a coral reef at Isla Larga, Venezuela. Sponges are important components of the benthic marine fauna with over 8,000 living species. They're known for their interactions with invertebrates seeking for food, shelter, and substrate for attachment. Among these groups, we found crustaceans, mollusks, ophiuroids, polychaetes, and fishes, which inhabit cavities and canals interiors. In the specific case of San Esteban National Park's coral reefs, sponges coverage is approximately 10%. We, in this study, described the macrofauna inhabiting Ircinia felix. This species represents a wide distribution along the depth gradient, being an ideal substratum for the invertebrates that inhabit these coral reefs. It also has a relatively small body size and an individual growth that facilitates its extraction and manipulation. Venezuela is located in the South Caribbean, and ISLA LARGA is located in the north coast, central north coast of Venezuela. Material and methods. We use 350-meter transects in 3 different depth strata, 1 to 3, 3 to 6, and 6 to 9 meters. We collected a total of 30 sponges, 10 per depth stratum, saved them in rotulated plastic bags, and transported them to the lab in ice coolers. Once in the lab, we proceeded to study the sponge's morphology, counting the number of oscula, size and shape, height, width, and length, color, volume, and if it was a low bait or a massive sponge. After this, we proceeded to extract the organisms that were in the sponge's tissues and to identify the associated fauna. As a result, we found over 60,000 individuals with a mean density of almost 12,000 individuals per liter. As we can see in the graph, there's no significant difference along the depth gradients, but we can see a slight difference in the mid-stratum between 3 and 6 meters, higher mean density. And also we can see with the coverage that in this same stratum, we have a higher coverage. And this can be because in the shallow area, the wave action is much higher, which decreases the sponge's ability to attach and grow, and mainly due to substrata instability and sediment particles' high resuspension rate. Also, in this specific coral reef, there's a high resuspension rate towards the deep stratum of the reef. We found a total of 9 associated taxa, being polychaetes the most abundant one, with over 98% of abundance. Here we can see the abundance of the polychaetes was pretty even along the depth strata, and also the Malacostraca and Cirripeda density. Anthozoas and gastropoda show a higher density in the shallow stratum. It's been commonly described that sponges that have a complex gross morphological structure often harbor a more abundant fauna. And it's been found in other parts of the Caribbean and Atlantic that sponges have a high number of and a high density of polychaetes, bryozoans, and nematodes. Now, in terms of polychaetes, we found a total of 16 families having the greatest diversity area in the 3 to 6 meters. In the shallow area, we found 11 families, and in the deep area, we found 10 families. Ciliates was the most abundant family in terms of density, with over 11,000 individuals per liter. And we can see it was also pretty even along the depth gradient. Among the other families, the most abundant ones were Sabellidae with 4.6%, Eunicidae, and Lumbrinaridae with 2.9 and 2.5 respectively. All other polychaetes occur in a very low density, which was lower than 1%. It's also been recorded in different studies that ciliates were the most common family and most abundant one, but also that the species Haplociliates spongicula was the most abundant one in general. In conclusion, we found that the density of taxonomic groups associated to Ircinia felix was slightly higher in the mid-stratum, where the total sponge coverage was higher. The most abundant taxa was polychaete, representing 98% of the total macrofauna. 16 families of polychaetes were identified. Ciliates were dominant in terms of density, followed by Sabellidae, Eunicidae, and Lumbrinaridae. Sabellidae, Eunicidae, and Lumbrinaridae represent high density values along the whole depth gradient, mainly on the shallow stratum. And the data presented here reiterates the proposal that sponges are important biodiversity reservoirs and they should be considered in conservation programs. Thank you very much, and I hope you enjoyed this presentation.